Hello everybody. We're going to do a little continuation with nucleic acids or DNA and RNA since it's been a while since we've seen each other, and we still needed to finish that unit before we left for spring break. So we'll do a little review on nucleic acids, DNA and RNA first, and then we'll get into the actual making of proteins and then continue on with the rest of our semester with our distance learning. Very exciting. All right. So remember, DNA and RNA are nucleic acids, which is a group of molecules they're really big. They're what we call a macromolecule. They're a huge molecule made out of repeating units, and those repeating units are called nucleotides. All right, so DNA, there's that long word, stands for deoxyribonucleic acid. And its job is to store our genetic information. And it looks like this picture here. Remember, it's twisted as a double helix, so it's like a winding staircase. And it's made out of these repeating units called nucleotides that only come in 4 different forms: the A, the T, the C, or the G. Adenine, thymine, cytosine, and guanine. At least in eukaryotes, we can find it in the nucleus of our cells. So here's a nucleus of an animal cell. It's inside there. And the nucleus of a plant cell. And then, of course, we haven't talked about this yet, but in eukaryotic cells, the mitochondria and the chloroplasts actually have their own DNA as well, and they actually make their own proteins, which is pretty interesting and leads us to other topics down the line on why that would be, why those types of organelles would need their very own DNA or their very own instructions to make proteins, because remember, that's what DNA does. It's basically an information or a recipe to be able to make proteins for the cell, because proteins are the molecules that basically build us, and those proteins can do so many different jobs in our bodies. It looks like this. If we did a flow chart, DNA, then we're going to need a secondary molecule as kind of a messenger in between, and it's called RNA. Very similar to DNA, but a little different. And then eventually what we want for the cell is to have proteins. We went through a bunch of different experiments back at about the turn of the century or so to try to figure out how do we know that it's actually DNA that's the genetic molecule. I mean, cells have so many different molecules. How do they actually know that it's the DNA and it's not like the proteins or something else? But one of the hot topics right now, of course, going on out there is viruses. So this is one experiment that helped us figure it out. It's called the famous blender experiment, where they were able to radioactively mark the DNA in these viruses here. These are called bacteriophages because they're viruses that actually attack bacteria. They look like little aliens, and so here's a diagram of them here. Basically, the virus has a protein coat with a little fatty layer underneath that protein coat, and then there's DNA inside here. Remember, some viruses might have the other genetic molecule called RNA, but in this particular case, it's DNA. And then we found a way to radioactively mark that molecule so that when these bacteriophages, these viruses, landed on bacteria, and then they basically squirted in or injected this material here, we could figure out that it's the radioactive stuff that we had marked earlier up in here, that they were actually putting their DNA into these bacterial cells. And then those cells of the bacteria would start making virus proteins, because basically it has the instructions to be able to do that now with the viruses putting their own DNA inside there. So it's a pretty famous experiment, but it helped to really confirm that it is really DNA, the genetic molecule. All right, so here's DNA, deoxyribonucleic acid, as a double helix, and here's its relative, probably the relative that came before DNA. It's called RNA, or ribonucleic acid. And you'll notice it's just like a single helix or a single staircase. One, deoxyribonucleic acid is what that one stands for, and then ribonucleic acid for RNA. Both of them are relatives of each other because they're made out of nucleotides, or those repeating units. So nucleotides have three main parts. All right, we'll take a look at that. So one of the parts is a sugar. So up in the upper right-hand corner are pictures of the two different sugars that make up DNA and RNA. You'll notice they're like really, really close to one another. They look very similar. When these sugars are in a watery environment, they curl into a pentagon format and look like they do in the picture. All right, then at each of these corners is a carbon. We just don't put it in there. We just understand that, okay, here's the number 1 carbon, the number 2, 3, 4, and then the 5th one, you can actually see where it is. So C is for carbon, and remember H is for hydrogen, and then O, oxygen. So basically sugars have a lot of those 3 elements: carbons, hydrogens, and oxygens. And then DNA's sugar deoxyribose is very similar to that. Except if you look in this little corner too, by the number 2 carbon in the lower corner, you'll notice that in ribose there was an OH, but in deoxyribose there's only an H. So do you think maybe that's how deoxyribose got its name? Because it's missing an oxygen there, whereas ribose actually has the oxygen? Could be. I'll let you check it out to find out. All right, so all nucleotides have a sugar. All right, then there's also a phosphate group, which is basically a phosphorus atom surrounded by some oxygens, as we see here. This phosphate group has generally a negative charge, which basically gives DNA a negative charge, which that becomes useful when we do things in the lab like DNA fingerprinting and trying to solve crimes. And then the part that's different with all the nucleotides is the base, or what we call a nitrogenous base. And there's, at least in DNA, there's 4 different kinds. And then in RNA, there's also 4 different kinds. They're all the same, except for the T is not found in ribose. Instead, there's one called U, or uracil. All right, so here they all are. I'll get my picture out of the way. And so here's what adenine looks like, or A. And the reason we call them nitrogenous bases, because they have a lot of nitrogen in them. N is for nitrogen. So adenine and guanine, you'll notice they look very similar. They have a pentagon and a hexagon near one another. At least in these diagrams, we can see the carbons. They're actually shown. And then the other nucleotides that are just a single, like a pentagon, we've got cytosine, or C, thymine, T, which is only in DNA, and uracil, a U, which is only in RNA. All right, take a look, if you want to pause the video, take a look and see if you can actually see the difference between thymine and uracil. There's not a lot of difference there between those two. You'll notice there's just a little CH3, that's called the methyl group. That's on thymine, but it's not on uracil. So these two very similar. One of them's in DNA only, and one is in RNA only. But adenine, guanine, and cytosine can be found in both DNA and RNA. All right, now I'm going to make this a little bit bigger, if I can. All right, we're going to do a little DNA building here. All right, so here we actually see the nucleotides, and the blue circles are circled around each nucleotide. You'll notice they're chained together. I can see the is it orange or yellow? That's the base. And the green is the sugar, and the little round circle is the phosphate group. Notice that the sugars on what we call the backbone are attached to the phosphate, and then our bases stick toward the middle. So then it looks like this. All right, so DNA we know is double-sided. It's a double helix. So the A's always match with the T's. Why? Because they like to have 2 hydrogen bonds between them. And then G's always match with the C's. Why? Because they like to have 3 hydrogen bonds between them. All right, that's actually kind of nice because, well, DNA is 3 billion of these long. Well, then broken up into our 46 chromosomes, but 3 billion long, that's a lot. So if we know one side of DNA, that means we can figure out the other side, because if I have a T on one side, I know there'll be an A on the other side. If I have a G on one side, I know, oh, there'll be a C on the other side. So that becomes very important. That's called complementary base pairing. So there's our DNA molecule. Looks very pretty. It's very small. These measurements are in nanometers, and nanometers are 10 to the negative 9, so it's really, really small. So those hydrogen bonds then join those 2 strands together. When DNA copies itself, as it has to do whenever mitosis happens, or meiosis, or binary fission in prokaryotes, whenever that happens, it's called replication. But it's okay. Replication can, well, I don't know if I want to say it's easy, but we can unzip these weak hydrogen bonds between our bases, and then we can fill in the middle. Because we already have our template that we know there's a G right here, well, I know then I would fill in a C as its complementary base. And when I have an A over here, I'd fill in a T as its complementary base. All right, so here's some times when DNA would actually need to be copied during cell reproduction or whenever we make gametes. And we'll send a little shout out to these gentlemen who figured out that structure of DNA. They actually went about it in an interesting way and put together research from different scientists to figure this out. James Watson and Francis Crick in 1953. Once they started figuring it out, they went and made this model here. Looks like it's made out of a bunch of wires and such. And they used some research with the help of Wilkins and Rosalind Franklin. And this is one of her X-ray crystallography pictures that helps mathematically tell us that DNA is a double helix. And on another interesting note, we've mentioned ATP before during our year. That stands for adenosine triphosphate. That's our main energy molecule in our bodies. Well, it turns out ATP is made out of pretty much the same stuff that DNA is made out of, and RNA. Actually, more close to RNA because it has the sugar ribose right there. It has adenine, just like DNA has adenine and RNA has adenine. And then there's 3 phosphate groups. In order to build DNA, actually when these nucleotides come to the molecule to get attached, they actually have the 3 phosphates on them, but the 2 get broken off, so then DNA and RNA end up only having the 1 phosphate group. But ATP has 3, and we'll talk later in the year on what ATP does. But I just wanted to bring up a quick little note that it's very related to DNA and RNA because of its makeup. Somebody had an awful lot of time on their hands here, all right, to build that. So nucleic acids function for information storage. The order of the A, T, C's, and G's is called our genetic code. And the order that they come in can help tell a cell how to build proteins, as we will eventually see. All right.